Welcome to Behind the Manuscript, 10-minute conversations with the Massachusetts General Hospital scientists about their recently published work. In these brief, yet engaging dialogues, you'll not only explore the science, but also get to know the fascinating individuals making it happen. So, grab a cup of coffee and let's get to it. Time for science. Hello, everyone. I am your host, Marcela Quintanilla, and in today's conversation, we're speaking with Dr. Wilfredo Garcia Beltran. Dr. Garcia Beltran is a physician scientist who is also an attending physician in transfusion medicine and clinical pathology at Massachusetts General Hospital. Additionally, Dr. Garcia Beltran is a principal investigator at the Reagan Institute. In today's conversation, we will be discussing Dr. Garcia Beltran's recent publication in the journal Cell titled Evasion of NKG2D-mediated cytotoxic immunity by cervical viruses. Welcome, Dr. Garcia Beltran. It is really a pleasure to have you here.
It's wonderful to be here. Thank you so much for inviting me, Marcela.
So why don't we start by talking about your journey? How did you get to MGH? How did you start at the Reagan? Tell me all about it.
Of course. So I was born and raised in Puerto Rico. There I actually had completed a college degree in chemistry where I had done some research experiences that exposed me to the magical world of science, while at the same time having a lot of interest in medical training, given that it's kind of a very common career path to take when you're doing a science degree. So when I was halfway through college, I decided that I didn't want to choose between science or medicine, and I decided to do both. And I applied to dual degree MD PhD programs in Puerto Rico and in the US. And I actually was invited to interview and eventually accepted in the Harvard MIT MD PhD program, which I started in 2010. That got me 8 years of medical and scientific training, some of it at MGH, given that a lot of my clinical rotations were at Massachusetts General Hospital, but also my scientific lab was in the Reagan Institute of Massachusetts General Hospital, MIT, and Harvard. And so I had a lot of MGH affiliations during my training, and I just loved it so much here. I didn't want to leave, so that when I graduated, I decided to pursue clinical training in clinical pathology and transfusion medicine here at MGH. And even after completing that, I am now here as an attending in the pathology department, seeing patients in the blood bank and also working on reports and pathology, but also at the Reagan Institute, running my own lab as a principal investigator, studying natural killer cell biology, all within the MGH network.
That is so wonderful. It's just an incredible journey that you have, and just thinking about like 8 years that you dedicated to all of that. That is so impressive. Now, just like diving deeper into this paper, can you tell me about the findings of this paper? What is new that you didn't know before? And, as lay-friendly as you can, looking at the paper, it took me a while to even be able to say the paper out loud.
Yes, of course. So I think the big thing that we found was a mechanism by which the virus is able to overcome our initial innate defenses against viruses. So for any virus or bacteria out there to actually cause disease in us humans, it needs to overcome our immune system and thus cause disease. Now, a lot of the studies around COVID-19 had been focused on antibody responses and also T-cell responses against the virus. But there's these initial responses by the immune system that happens within the first minutes to days that is really important for the virus to overcome to cause disease. And so one of the cell types that the body has as the first line defense against viral infection are called natural killer cells. So natural killer cells are a kind of immune cell that defends the body from viral infections. And so we wanted to understand in this study, how are these cells recognizing infected cells, and what are infected cells doing to prevent being eliminated in that first initial infection that then causes actual disease. So what we found in this paper is that SARS-CoV-2, the virus that causes COVID-19, is able to evade recognition by natural killer cells, that first line defense, by kind of shedding or releasing these signals present on the surface of infected cells that normally signal to natural killer cells that this cell is infected and must be killed. So the virus is able to kind of make the cell it's infecting kind of blind to NK cells. And so by identifying that mechanism, we kind of went further to see, can we stop the virus from doing this? And in fact, we found a medication that's used in the cancer field that can prevent the virus from shedding these signals that tell NK cells to kill the infected cells, thus allowing us to enhance killing and elimination of infected cells. So I think these two findings were kind of pivotal moments where we understood how is SARS-CoV-2 overcoming this natural killer cell immunity to cause disease in humans as it did during the pandemic, and what can we do to stop the virus from doing that so we can better combat it.
That's so interesting. And it completely started in your lab, right?
Yes. So it's always a collaborative effort. I love that about science. Science is never done in an island. It's always done in collaboration with other teams. So this project was a very fruitful collaboration between my lab and the co-director of the BSL-3 core at the Reagan Institute, Julie Bucco. So she is an expert in the culturing and, experimental setup for these very kind of risky pathogens. And so that's why we have special facilities to work with live virus. My lab focused a lot on the NK cell biology aspect, and her core facility was focusing on being able to actually use live virus. And so bridging those efforts, we were able to produce this publication that had started within my laboratory, authored by Jordan Hartman, who was a master's student at the time that was very interested in studying SARS-CoV-2 infection in humans.
So now I want to talk about the next steps. Where are you hoping to go from here? What comes next in your research?
That's a great question. So we currently have two avenues that we're pursuing. One of them is more on the basic science fundamental side, where we actually want to find the mechanism by which SARS-CoV-2 is able to induce the shedding and evasion from natural killer cell immunity. And so we currently know that there's a viral protein called ORF6 that's responsible for the shedding phenomenon. That protein is actually shared with other viruses in this virus family called sarbecoviruses. And those are viruses that have caused outbreaks in the past and may cause future pandemics. And so we're hoping that by uncovering the mechanism by which SARS-CoV-2 and the related viruses are able to induce the shedding phenomenon and evade immunity, we think we could create then an avenue for the development of small molecule inhibitors that can block this viral protein from its immune evasive effects, and that way have a drug that can cover multiple viruses, not only the current one, but potentially future ones. On the other side of things, we have a more translational aspect that we're pursuing, which is with the medication we have on hand, so the monoclonal antibody 7C6 that blocks shedding of these signals on the surface of infected cells, can we use that currently to treat patients with severe COVID-19? And so we're currently pursuing animal models where we infect animals with SARS-CoV-2 and then treat them with 7C6 to see if we can prevent severe disease, enhance viral clearance. And if that proves to be true, we can then proceed to kind of testing this in patients in clinical trials and seeing if we have a novel form of immunotherapy that originally was used to treat cancer but can now be used to treat a viral infection.
I know that, you've mentioned that you're still steps away from patient care, but I do want to ask, how do you think that this work can potentially impact patient care?
Wonderful question. So in a very focused manner, we believe that the medication we have on hand, which is a monoclonal antibody that actually blocks the shedding phenomenon, can serve as a form of immunotherapy for severe SARS-CoV-2. So if we can show in animal models that this medication can prevent severe disease, we can then move into humans and use it to treat severe infection, which is now much less common due to vaccines, but still occurs. However, in a more broader sense, the idea of using immunotherapy to treat a viral infection is a quite novel concept, where normally we think of immunotherapy in the cancer field, and that's where this medication came from, so we can modulate the immune system to kill off the cancer. But now we're looking at, even in the context of a viral infection, we can also, in that setting, modulate the immune system to clear the virus more efficiently and prevent severe disease. So we think that's how we can, in maybe 5, 10, 15 years, affect patient care and the way medicine practices in the context of infectious disease.
And now just going a little bit more into just learning about you, if you weren't doing science or medicine right now, what do you think you would be doing?
It's a great question. I've always had a very artsy side. I've been a big fan of music. I've actually been part of a cappella choirs ever since I was in high school, college, medical school. I even formed the choir during my PhD, and directed it last year, even as a principal investigator. So if I couldn't do science or medicine, I would probably be creating music, singing as part of choirs, and just kind of pursuing that career.
Thank you so much, Dr. Garcia Beltran. I really enjoyed this conversation with you so much, and I'm sure that many people will be fascinated about your research. And I hope that we get the chance to have more conversations like this and about your science in the near future.
Thank you so much for having me, Marcela. It was a pleasure.